Hello students, welcome to GDC Live. So in the last class we were talking about glycolysis. We learned about what is glycolysis basically means. Means oxidation of glucose to pyruvate and lactate, and by this process energy is generated. Okay, so we talked about what are the different enzymes involved, what are the byproducts involved, how many energy are the form of ATP generated. We have discussed in detail. In the today's video, we wanna talk about the rest of the oxidative pathway of glucose. So in the today's class, we will start with TCA cycle. So as you know that TCA is also called as tricarboxylic acid cycle. It is also known as tricarboxylic acid cycle, or also known as Krebs cycle, because this cycle is basically concept given by Hans Krebs, one of the scientists. By his name, we call known study this cycle as a Krebs cycle. So this is also known as citric acid cycle, Krebs cycle, tricarboxylic acid cycle. It is the most important metabolic pathway for the energy supply to the body and operate in aerobic condition only. This is very important to remember. So as we told you in glycolysis, what basically happens is this was carried out in both aerobic and anaerobic condition. In aerobic condition, uh, we get pyruvate, whereas in case of anaerobic condition, we get lactate. So in aerobic condition, total number of ATP we net ATP we gain is 8. So in case of lactate, we get 2 ATP. So with oxygen, without oxygen. Okay, but this tricarboxylic acid cycle, this is only occurs in Krebs cycle. And what is another difference? We learned that glycolysis is occurs in, this is carried out in cell cytoplasm. This is carried out in cell cytoplasm. But this TCA cycle is carried out in mitochondria. This is carried out in your mitochondria. So around 60 to 70% of the ATP is synthesized by Krebs cycle. So you can see what is the importance of Krebs cycle from this number. Around 65 to 75% of your total ATP is synthesized by your Krebs cycle. Citric acid cycle is basically क्या होता है इसमें ऑक्सीडेशन ऑफ, वहां पे किसका ऑक्सीडेशन हो रहा था, वहां पे ऑक्सीडेशन हो रहा था ग्लूकोज का. So ये जो ग्लाइकोलिसिस हो रहा था बेसिकली द स्टार्टिंग मटेरियल इन ग्लाइकोलिसिस वाज बेसिकली योर glucose C6H12O6, your glucose molecule. Here your starting material is your acetyl coenzyme. Now you will tell, sir, where the acetyl coenzyme is comes from. I will tell you in the upcoming slide where this acetyl coenzyme basically comes from. From this pyruvate, how the acetyl coenzyme formed and how this process will carried out will be discussed in the upcoming slide. So acetyl coenzyme converted to your carbon dioxide and water. So basically इसमें क्या होता है, there is a oxidation of your acetyl coenzyme and due to the oxidation there is a formation of your carbon dioxide and water. The final and common, this is the final and common oxidative pathway for the carbohydrate, final and common oxidative pathway for your carbohydrates and fats and amino acid. This is the final and most common oxidative pathway for the carbohydrate, fats and amino acid. Now, the Krebs cycle is the most important central pathway connecting almost all the individual metabolic pathway directly. What it is saying is, so central जो आपका TCA cycle है और Krebs cycle है, this is connecting to other metabolic pathway either by directly or by indirectly, but it is connecting. Okay, it is connecting to other pathway. The enzyme of TCA cycles are basically located in the mitochondrial matrix. That's why this process or this cycle basically carried out in your mitochondrial matrix. So basically it is carried out your mitochondrial matrix. It is occurs only in aerobic condition. Around 65 to 70% of your ATP will be produced from this TCA cycle only. What basically TCA cycle is? There is the oxidation of your acetyl coenzyme A to carbon dioxide and water, and the final product, final common pathway, this is the final and common pathway for the metabolism of carbohydrate, fats and amino acids. Okay, let's understand this cycle in detail. Okay, so see how this pyruvate, so this pyruvate is obtained from glucose by the process, कौन से प्रोसेस में? Glycolysis. By the process of glycolysis, glucose will give pyruvate, and from this pyruvate, acetyl coenzyme will be generated. What will generated? Acetyl coenzyme. In the presence of an enzyme called pyruvate dehydrogenase. So, in the presence of enzyme called pyruvate dehydrogenase, so the acetyl coenzyme will be produced from pyruvate. Now you can see the series of reactions. So what basically happened? So this acetyl coenzyme, what it is do is it convert oxaloacetate to citrate. What would basically do is it causes oxaloacetate to citrate. So in the presence of an enzyme called citrate synthetase. So in the presence of enzyme, जो enzyme citrate synthetase है, in the presence of the that acetyl coenzyme may convert oxaloacetate to citrate. Now this citrate will convert it to cis aconitate. Citrate will convert it to which compound? It will convert it to cis aconitate in the presence of a enzyme called aconitase. See, so what basically it is occurring? Citrate is converting to cis aconitate in the presence of aconitase, and here H2O is releasing. So that means there is a hydrolysis takes place. Okay. Now cis aconitase again converted to isocitrate. Cis aconitase is again converted to isocitrate. Again releasing of your water molecule. Enzyme will be both same. Now, isocitrate is converted to oxalosuccinate. Isocitrate is converted to oxalosuccinate. See, here your NADH is released. That means your energy production started in the presence of isocitrate. As I discussed you, 1 NADH is equal to 3 ATP, 1 FADH equal to 2 ATP. Remember, so that will be help you for the calculation. So 1 NADPA is you give 3 ATP, 1 FADH is. See, from the conversion of isocitrate to oxalosuccinate, there is a production of ATP in the presence of isocitrate dehydrogenase. Now, this oxalosuccinate will convert it to alpha ketoglutarate. Oxalosuccinate will be converted to alpha ketoglutarate. Here you see there is a release of carboxylation. So D is this process is called as decarboxylation. Okay, so here carbon dioxide is released in the presence of isocitrate dehydrogenase. Again, alpha ketoglutarate is converting to succinyl coenzyme. Here also carbon dioxide release. Along with that, there is a release of energy. 2 NADPH. So 1 NADPH is here, another NADPH is here. Now, from succinyl coenzyme to succinate, your enzyme is succinyl thiokinase. Here you can see that 1 GTP is produced. Okay, now from succinate to fumarate, 1 FADH is produced. So see, and from fumarate to lactate, and from lactate to again oxalosuccinate, oxaloacetate, your 1 NADPH is produced. So see how the energy production basically here 1 NADPA, see the energy, how this energy generating process basically takes place. So I am again repeating. So from the glucose we get oxidation of glucose, we get pyruvate. From the pyruvate converted to acetyl coenzyme by presence of a enzyme called pyruvate dehydrogenase. Here also you will get one NADPH. Okay, now this acetyl coenzyme, what it will do is it will convert oxaloacetate to citrate in the presence of an enzyme called citrate synthetase. So in the presence of enzyme called citrate synthetase, oxaloacetate basically converted to citrate. Now this citrate converted to cis aconitate in the presence of an enzyme called aconitase along with release of water molecule. Now the cis aconitate converted to isocitrate in the presence of same enzyme called aconitase. Now isocitrate will converted to oxalosuccinate. Here you will get an energy. Now from oxaloacetate to alpha ketoglutarate in the presence of enzyme isocitrate dehydrogenase. Alpha ketorate converted to succinyl coenzyme in the presence of enzyme alpha keto dehydrogenase. If from succinyl coenzyme, it converted to succinate in the presence of enzyme succinyl thiokinase. Succinate converted to fumarate in the presence of enzyme succinate dehydrogenase. Question पूछता है कि at what stage your FADH is produced. So from the succinate to fumarate, F से याद रखना जहाँ से fumarate produce होता है, in that stage only FADH is produced. So fumarate to lactate and lactate to oxaloacetate. So this is called as TCA cycle. This basically known as your TCA cycle. Okay, so let's see the energetics. So how energy is produced. So you can see during the process of oxidation of acetyl coenzyme high citric acid cycle, for reducing equivalent 3 as a NADPH आपको देखने के लिए मिल रहा है 3 NADPH and 1 FADH are produced. So oxidation of 3 NADPH will lead to 9 ATP, whereas FADPH lead to 2 ATP. Other than that, you will get 1 GTP here. So number of total NADPH is 1 here, 1 here, and 1 here. 3 NADPH, 1 FADPH, 1 FADH, 1 GTP. So total of 3 NADPH means 9 ATP. 1 FADH means 2 ATP, 1 GTP, it will calculate as 1 ATP. So total number of ATP is called as 993, 93 add किए तो 12 ATP. So total number of ATP produced from one citric acid cycle is 12 ATP. Okay, so 12 total number of ATP that produce is 12 ATP. So this table basically show the action of vitamins over your citric acid cycle. Thiamine, your vitamin B1, your vitamin B1, what basically it is a coenzyme for alpha ketoglutarate dehydrogenase. So thiamine basically acts as a coenzyme for your alpha ketoglutarate dehydrogenase enzyme. So thiamine basically alpha ketoglutarate dehydrogenase enzyme, where it is basically act. See, this is the enzyme. So if for this thiamine act as a cofactor. Similarly, riboflavin is the coenzyme for succinate dehydrogenase. Riboflavin is for the coenzyme of succinate dehydrogenase. Riboflavin is your vitamin B2. Riboflavin, vitamin B2. Similarly, niacin is a electron acceptor for isocitrate dehydrogenase. It is a electron acceptor for isocitrate dehydrogenase, alpha ketoglutarate dehydrogenase, and malate dehydrogenase. So this is coenzyme. Niacin basically act as a your electron acceptor for isocitrate dehydrogenase, alpha ketoglutarate dehydrogenase, and malate dehydrogenase. Pantothenic acid is basically your vitamin. Which one? Vitamin E B5. Niacin is vitamin B3. Riboflavin is vitamin B2. So pantothenic acid attached to the active carboxylic acid residue acetyl coenzyme and succinyl coenzyme. This is the action of your vitamin B complex over your TCA cycle. So let's see the inhibitor. So exam asked from this inhibitor. So what are the inhibitor basically available for the different enzymes? So these inhibitors are what basically do is these can block the action of this particular respective enzyme so that they can inhibit or stop the TCA cycle. Okay, so you can see aconitase inhibitor is fluoroacetate. Aconitase inhibitor is fluoroacetate and it is a non-competitive inhibitor. It is not a competitive inhibitor, rather it is a non-competitive inhibitor. Now alpha ketoglutarate dehydrogenase. So alpha ketoglutarate arsenite is basically considered as a alpha ketoglutarate dehydrogenase inhibitor, and malonate is a inhibitor of succinate dehydrogenase. Okay. So you have to remember that fluoroacetate basically act as a inhibitor of aconitase enzyme. Arsenide basically act as a inhibitor of alpha ketoglutarate dehydrogenase. Malonate is act as a inhibitor of succinate dehydrogenase. And both this malloate is a competitive inhibitor. Okay, so Krebs cycle is a amphipathic cycle. So amphibolic cycle. Remember आपको एग्जाम में क्वेश्चन पूछा which of the following is amphibolic? Why? Because Krebs cycle have both catabolic and anabolic function. TCA cycle is actively involved in gluconeogenesis, transamination and deamination process. This is also important that your TCA cycle is directly regulate the gluconeogenesis, transamination and deamination. You can see, so from the oxaloacetate, by the process of transamination, aspartate is formed, and aspartate is a non-essential amino acid for the purine and pyrimidine synthesis. Oxaloacetate by pyruvate carboxylase, pyruvate is formed, and from the malonate, pyruvate is formed by malic enzyme. Succinate, from the succinyl coenzyme, your heme can be formed. From alpha ketoglutarate, by process of transamination, your glutamate is formed, and you know glutamate is a non-essential amino acid and for purine synthesis involved. And your citrate is basically involved in your fatty acid and sterol synthesis. Here from this particular diagram, you can see how the TCA cycle is involving through different other synthetic process. I hope everyone is clear, getting my point. So I am trying to explain that this TCA cycle or citric acid cycle can be linked to other cycle like your gluconeogenesis. Gluconeogenesis means there is a synthesis of carbohydrate from synthesis of glucose from the non-carbohydrate cycle, like your glutamate. Glutamate is from glutamate also your glucose synthesis occur. That process basically called as gluconeogenesis. Like your transamination, by process of transamination you can see it is linked, TCA cycle is linked, and by the process of transamination your aspartame and glutamate can be produce. Okay, so that's all about your TCA cycle. Now we will discuss in detail about gluconeogenesis. So what is gluconeogenesis in a layman language? I can tell you that gluconeogenesis is synthesis of glucose. Gluconeogenesis, glucose what is there na, देखो glucose what is there, glucogenesis what is there, means glucose, that means glucose का तो synthesis हो रहा है, but neogenesis means synthesis of the glucose from non-carbohydrate compound. Carbohydrate compound से ना होके it is basically synthesized from a non-carbohydrate compound. That process is basically called as your gluconeogenesis. So this is a major substrate or precursor for glucose. So what are the main question asked in different exam? What are the precursor or substrate for gluconeogenesis? That means from which molecule your gluconeogenesis process can be carried out, like your lactate. You know lactate is synthesized from your glucose by the anaerobic condition, from the lactate, even from the pyruvate, glucogenic amino acids, propionate and glycerols. These are basically act as a substrate for the gluconeogenesis. This is very very important from the exam point of view. So you can get a question which of the following, like you can get a question like which of the following is not a substrate for uh, gluconeogenesis, like that also you can get a question. This is basically gluconeogenesis, like mitochondria. In mitochondria, I told you that TCA cycle is carried out in mitochondria. Similarly, this gluconeogenesis basically carried out in your cell cytosol, although some of the precursors are produced in mitochondria. So gluconeogenesis, basically the major location of gluconeogenesis is your liver. This is very important. You can remember. So liver is the major site for the gluconeogenesis. This is the major site. Okay. So what is the importance of glucogenesis? Why this gluconeogenesis is important? Remember, the importance of gluconeogenesis is basically in case of fasting condition. So like whenever you go for a fasting and for prolonged period, for prolonged time, like 1 or 2 days, um, what basically occurs is your fat started burning. That's why nowadays people are doing fasting. So what basically intermittent fasting is, you will give 12 hour of gap between 12 hour of gap between two major meal. Okay, so what basically occurs in that case is, in that 12 hour your fat will started burning, because in our body, our normal glycolysis process, any metabolic process will be carried out. That process won't be stopped. For carrying that particular process, we need ATP. Obvious thing that we need ATP or energy. Now we are not taking carbohydrates, because carbohydrate is the major source of energy. So what body will do is, it will synthesize ATP or energy from non-carbohydrate sources like your propionate, lactates. In that cases, fat also act as a source, because lipids, your amino acids are different. These are act as a source. So in that cases, your fats are started burning. So that is a good technique for weight loss. Okay. So brain and central nervous systems, erythrocyte, testis, kidney, medulla, and depend upon the glucose for continuous supplies of energy. Human brain alone require 120 gram. हमारा brain जो है alone it requires 120 gram of glucose per day, about out of 160. Every day our body needs 160 gram per day glucose. Okay, from that 160 gram, 120 gram will be used by your brain. This is your whole body need. This is your whole body's need. So 120 gram is only your brain's need. Okay. Next is glucose is the only source of supply of energy in skeletal muscles under anaerobic condition. So in fasting condition more than a day, gluconeogenesis is most occurs to meet the basal requirement of the body for glucose and to maintain the intermediate citric acid cycle. This is essential for the survival of human and other animals. So this is basically important for the survival of human and other animals. Certain metabolic products in tissue, certain metabolic products in the tissue basically accumulates in the blood. Example, your lactate, your glycerol, propionate, etc. Metabolic products in the tissue, like these are has to be removed, like lactate, glycerol, propionate. So these basically removed from clear from the blood by the process called gluconeogenesis. By the gluconeogenesis, this basically cleared from the body. Next is your how gluconeogenesis process basically takes place. So remember, gluconeogenesis basically the reverse reverse process of gluconeogenesis, basically your reverse process of which reverse process? This is the reversible process of or glu glycolysis. So we can say the gluconeogenesis is just opposite of your glycolysis. So you can see, in if I will come from top to bottom, from glucose 6 phosphate, from glucose 6 phosphate to fructose 6 phosphate, fructose 1 6 phosphate, 1 3 bisphosphate, 3 phosphoglycerate, 2 phosphoglycerate, phosphoenol pyruvate or pyruvate. So this basically carried out basically in which process? This basically glycolysis. So glycolysis means your oxidation of glucose. But here you can see this is just reverse. So but the reversible process, how it start? Your pyruvate converted to oxaloacetate, then the process gets start directly. Here one intermediate is there, oxaloacetate. So oxaloacetate comes means there is a involvement of TCA cycle. As I told you na, TCA cycle is also here. I mentioned one point that TCA cycle is also actively involved in gluconeogenesis. This oxaloacetate, this oxaloacetate produce pyruvate. Now from this pyruvate your gluconeogenesis will start. From this pyruvate your gluconeogenesis will be start. You can see how this pathway are interlinked, how this pathway are interconnected with each other. See, this pyruvate converted to your oxaloacetate. Now from the oxaloacetate converted to phosphoenol pyruvate to 2-phosphoglycerate, 2-phosphoglycerate to 3-phosphoglycerate, 3-phosphoglycerate to 1,3-bisphosphoglycerate, to glyceraldehyde 3-phosphate, and to fructose 1,6-phosphate, and your glucose will be synthesized. So this process basically carried out in your liver. This is a short diagram that from the lactate, in the presence of lactate dehydrogenase enzyme, pyruvate is produced. From pyruvate, in the presence by the process gluconeogenesis, your glucose is produced, and this process basically takes place in your liver. I hope everyone is clear up to this about gluconeogenesis. So next is Cori cycle. Cori cycle is basically a variety of gluconeogenesis. So this is the synthesis of glucose from lactate by the process of gluconeogenesis. If the glucose synthesis, I have shown you that from pyruvate your oxaloacetate is synthesized. So if the synthesis is start from lactate, okay, if the synthesis is start from the lactate, then it is called as gluconeogenesis. This cycle involves synthesis of the glucose in the liver from the skeletal muscles lactate, and the reuse of glucose thus synthesized by energy purpose known as Cori cycle. So basically देखो क्या होता है, I'm explaining you. In muscle, you know, muscle is skeletal muscles is in skeletal muscle, when glycolysis takes place, this is your skeletal muscle. So skeletal muscle, you know, you have to like when you have to walk, you have to like when right now I am teaching you and you are learning. So your skeletal muscle is involved. Like you are writing, you are seeing, and you are changing your page, turning your books, seeing your books. All these cases, your skeletal muscle is involving. In that skeletal muscle involvement process, your ATP will be utilized. So by the process of glycolysis, and you know, in if an anaerobic condition is there, oxygen sufficiently not available, then your lactate is synthesized. Now this lactate will enter into your liver. Lactate will enter into where? Your liver. And from the liver, by the process of gluconeogenesis, your glucose is formed. This cycle basically called as Cori's cycle. This is very important from the exam point of view. So if will question us, Cori cycle basically takes place in which organ? Specific पूछा, तो that will your answer will be liver. Liver is the major site where lactate converted to glucose, and that lactate basically produced in your skeletal muscle. I hope I am clear. I'm again summarizing that Cori cycle means synthesis of glucose from the lactate by process of gluconeogenesis. Where this lactate, if question asked, where this lactate you get? So this lactate basically obtained from your skeletal muscle, where glucose is metabolized. By the metabolism of glucose in anaerobic condition, your lactate is formed. Now this lactate is transferred from your skeletal muscle to your liver, and where from that liver, by the process of gluconeogenesis, first lactate is converted to pyruvate. Now from the pyruvate, your by the process of gluconeogenesis, your glucose is formed. That process is basically called as gluconeogenesis. I hope I am clear up to this. Next is your glycogenesis. There are two terms. One is your glycogenesis, another one is your glycogenolysis. Glycogenesis means synthesis of your glycogen, whereas glycogenolysis means breakdown of glycogen. So this is the synthesis of glycogen, glycogenolysis, synthesis of glycogen. This is not This statement is partially in wrong. The synthesis of glycogen from glucose is called as synthesis of glycogen from glucose is called as synthesis of glycogen from glucose is called as glycogenolysis. The degradation of stored glycogen in the liver and muscle is called as glycogenolysis. So in the glycogenesis, basically it takes place in their cytosol and requires ATP, UTP besides the glucose. So glycogen is degraded by breaking alpha 1,4 and alpha 1,6 glycosidic bond. Here you can see glycogenesis is carried by basically in the liver and muscles. So your ATP is utilized, your final ATP is utilized in glycogenolysis, your degradation of glycogen to produce glucose. Okay, G6P from glycogen, G6 glucose 6 phosphate is produced, and the from glucose 6 phosphate glucose and glucose is produced, and from the glucose 6 phosphate fructose 6 phosphate and glycolysis is produced in your skeletal muscles. So from the glycogenolysis, the glucose 6 phosphate is the common intermediate. From glucose 6 phosphate, glucose is produced in your liver and kidney, and in your skeletal muscle, glucose 6 phosphate converted to fructose 6 phosphate, and that fructose 6 phosphate basically involved in glycolysis process for the generation of enzyme, a generation of energy in your skeletal muscles. This is glycogenesis and glycogenolysis. So last one is, what are the disease that linked with this different type of glycogen? So we called it as a glycogen storage disease. So glycogen storage disease made, first one is type 1 glycogenesis, also known as von Wright disease. This question once asked in exam. Von Wright disease is basically deficiency of which enzyme? Glucose enzyme defect is glucose 6 phosphate. Glucose 6 phosphate. So this basically organ, what are the organs that involved in this particular disease is your liver, your kidney, and your intestine. So characteristics picture is glycogen accumulate in the liver, hepatocytes and renal cells, enlarged liver and kidney, fasting hypoglycemia, lactic acidosis, hyperlipidemia, ketosis, and gouty arthritis. This is the consequences and or we can say the characteristics picture. So von Wright disease is basically due to depends in which enzyme, that is glucose 6 phosphate. So which organ basically involved? Your liver, kidney, and intestine. What are the features? Your glycogen accumulate in the liver cells and renal cells, and enlargement of hepatocytes and renal and uh, hepatocytes and kidney cells. Okay, next is Pom's disease. So this is basically due to enzyme defect of lysosomal alpha 1 for glucosidase. So basically due to deficiency of enzyme lysosomal alpha 1 for glucosidase or acid maltase. So in organs involved is all organs of your body. So glycogen is accumulated in the lysosome in almost all the tissue. Heart is mostly involved. Pom's disease में your heart is mostly involved. Enlargement of liver and heart. Nervous system also affected. Death occurs at early stages due to heart failure. So major question आपको पूछ सकता है कि death in Pom's disease is basically due to uh, death in Pom's disease is basically due to the heart failure. Next is Coris disease. Coris disease is limit disease, also known as limit dextrosis or Forbes disease. So this is the deficiency of enzyme defect in amylose alpha 1,6 glucosidase, which is a branching enzyme. Amylose alpha 1,6 glucosidase. So organs involved is your liver, आपका liver, muscle, heart, and leukocytes are involved. So branch chain glycogen accumulate, liver get enlarged, that is hepatomegaly. Enlargement of liver is known as hepatomegaly. Okay, but milder compared to the von Wright disease. Then Anderson's disease, also known, this is the deficiency of glucosyl 4,6 transferase, which this is also a branching enzyme. Almost all of your tissue is involved in this disease. A rare disease, glycogen with only branch accumulates, cirrhosis of liver, impairment of liver function. So majorly your affected organ is here, your liver. So you can see most of the glycogen storage disease cases, your liver is affected because liver is the storage site of like a bank. Liver is a bank, like we deposit or we save our money in the bank. Okay, similarly, the extra money that what we are having left after the expenditure, we what we do is we kept in bank for the saving. Similarly, after the utilization of glucose, the extra glucose uh, that left after the utilization converted to glycogen, and that glycogen is stored in the liver. Liver is here is a type A bank. Okay, then McArdle's disease. This is a type 5 glycogenolysis. Muscle glycogen phosphatase enzyme, phosphorylase enzyme is involved. Basically, your skeletal muscle is involved. So, muscle glycogen is stored very high, not available during exercise. Okay, then Hair's disease is the liver glycogen phosphorylase. Your majorly organ involved is liver. Your liver is enlarged. Tauris disease is a deficiency is defect in the enzyme known as phosphofructokinase. That is your skeletal muscle. Majorly your skeletal muscles and erythrocytes are involved. So muscle cramps due to exercise, blood lactate, muscle blood muscle cramps due to exercise, blood lactate not elevated, hemolysis occurs. Okay, so in this figure I have shown you where this particular enzyme. So first is hexokinase or glucokinase. I told you this is von Griggs disease. So von Griggs disease is involvement of glucose 6 phosphate. So type 1 type of, so glucose 6 phosphate deficiency leads to which disease? Von Griggs disease. Next is type 2. Type 2 was your Pom's disease, lysosomal 1 for glucosidase. So type 2 is this is due to this is due to on branch of alpha 1. This is due to, and this lysosomal alpha glucosidase dysregulation can generate your Pom's disease. Then third one is your Coris disease, and Coris disease is basically due to amylase alpha 1,6 glucosidase. So this is a debranching enzyme. So this step this regulation leads to Coris disease. Okay. Then type 4. Type 4 is basically due to your Anderson disease, your glucosyl 4,6 transferase. So type 4 here glucosyl 4,6 transferase. This is, so its dysregulation or defect can leads to Anderson's disease. Then type 5 and type 6. Type 5 and type 6. Uh, McArdle's disease and Hare's disease. McArdle disease and your Hare's disease. Okay, and your type 7 from fructose 166 phosphofructokinase is basically involved. This is the deficiency in this enzyme leads to Tauris disease. So all these I have discussed is basically due to deficiency of this particular disease. So let me summarize what we have read today is. So we basically start from TCA cycle. We'll see that TCA cycle there is the oxidation of your acetyl coenzyme A to carbon dioxide water, and during this oxidation process we get energy. Total number of ATP generated from one TCA cycle is 12 number of ATP. Okay, so next we learned about what are the different intermediate forms, what are the enzymes involved. We also read about the effect of different vitamin B complex over our TCA cycle. We also learned about what are the inhibitors, like aconitase inhibitor is fluoroacetate, alpha ketoglutarate and dehydrogenase inhibitor is your arsenate, succinate dehydrogenase inhibitor is your malate. And I also explained you how this TCA cycle is linked to gluconeogenesis and other pathways. Next, we learned about gluconeogenesis, how this non-carbohydrate substrate like your lactate, pyruvate, glucogenic amino acid, propionate, and glycerin involved in synthesis of glucose. This is majorly synthesized in a liver, and this is basically the major importance is in case of starvation condition or fasting condition. And another one is the metabolite that are generated like your lactate, glycerol, propionate, that also eliminated from the body by means of gluconeogenesis. Now I also explained that gluconeogenesis is just opposite of glycolysis. There is but there is a intermediate form, that is oxaloacetate. Okay, next we learn about Cori cycle. Cori cycle is synthesis of glucose from the lactate in liver is called as Cori cycle, and this lactate basically obtained from the metabolism of glucose in skeletal muscle by the process of glycolysis in anaerobic conditions. Next we talk about glycogenesis and glycogenolysis, how this glycogen basically synthesized from glucose, and how the breakdown of glycogen can leads provides us energy by form of glucose. Then we learned about what are the different disease like your von Crick's disease, Pharm's disease, Colles disease, Anderson disease, Macadel's disease, Harris disease, Tawil disease. This table is 100% important from the exam point of view, because you can get question every alternative year से this table से question आता है. So this table is very very important. What you have to remember is the name of the disease, what is the enzyme defect. So name of the disease, what are the enzyme defect, and what are the majorly organs involved. Characteristics features is not that much important, but your disease name and what the enzyme involved, that is very very important from the exam point of view. Okay, so we have completed the carbohydrate. Okay, that's all for today. Thank you.